Cieplak from Institute of Physics in Warsaw. He'll tell us about nanoindentation of viral capsids.
Thank you very much. Well, I was wondering, that doesn't seem to be, it's working. I was thinking what to speak about, I mean, a week ago, 10 days ago, and there's a variety of subjects I could consider. But then I was inspired by this leaflet I saw at the Institute, which says that there's about this interdisciplinary approach to virus structure and assembly. Let me just make sure that this is on. Is it?
Seems to be. Nope. Should show red or green.
Okay, so if I press it. Oh, no, it's red. That's worse, right? It's either red or nothing. So
Down to the second top.
which one should it be, red or? Okay, thank you. So I've seen that the virus capsids have been already worked on and displayed. So I thought, okay, so maybe this would belong. And also the story I would like to tell you would tie to my lecture on stretching of proteins last week, because it's basically the same numerical model that I'll be using. But another thing that caught my attention when looking at this leaflet, and also several trips to Grantchester Orchard, made me think about apples. And another thing was that I just read that Apple filed a complaint accusing A.PL, PL is for Poland, of copying an Apple icon for its logo. This is a grocery, internet grocery place. So I thought maybe give you some story about apples first. And in particular, the question is, where are the British apples now? I mean, the falling apple started modern science in this place. So, and it turns out that, if you take the 10 biggest producers of apples in the world, then England is, or UK is not on the list. Well, the ranking depends on whether you measure it by weight or or value. The French apples are the most expensive, for instance. But also it depends on the source and the year. So this data for 2004. But if I look at the USA Today, which I believe was like 2 or 3 years later, then it lists, gives some different ranking and different numbers. So I don't know really which source to believe in. But whatever source you take, UK is not there. So that's a state of worry, 300 something years after Newton. Now, so I'll give you a picture of Polish apple, and it's easy. And it's easy to manipulate apples because you can just squeeze them, eat them, or whatever. But I'll be interested in squeezing objects that are like 200 angstroms across, like virus capsid especially. So there are these new tools to infer conformational changes in biological systems and study them mechanically. And one of them is atomic force microscope. And you can use it, for instance, to stretch proteins. If you can anchor one end and pull by another, by the tip of the AFM, then you can start the elastic response, which is first Hookean, and then it becomes complicated. And I was telling you about this last week. But the interesting feature here is to study, one of the interesting features is to study the characteristic scale of mechanical response, which we denoted by Fmax. But you can also use AFM to squeeze objects. If you anchor, small capsules of something, they also do, it could be even a red blood cell, but here it's a virus capsid, and you can take a thicker tip, you mount a thicker tip on the lever, and then you can press on this and then see what is the reaction to indentation, or in this case called, nanoindentation. So let me go back to proteins first, because
Is the tip fairly like a hammer, like a ball hammer? It's round.
Well, Yeah.
It's more rounded, this one.
Oh, okay, so there are all kinds of tips. You see, depending on the purpose, they might be very thin or broad, sturdy or weak. I mean, there's a variety of tools, okay, depending on the purpose. For viruses, you'd rather want to take, you want to take broader tips, like 30 nanometer, in curvature. But
Can you just also explain what you mean by a virus capsid for people that are not in?
I will.
Oh, you will.
I will. But before I do that, I realize that, I understand this is interdisciplinary, that's why I added the economical aspect to it as well.
Gastronomical.
But basically capsids are made of proteins, so let me say a few words about proteins first. So if you take this, this is what I was showing you last week, so if you take a well-studied protein like titin, then when you stretch it, it has this force peaks, and this characteristic scale for this of order 200 piconewtons, just to give you a scale. And then if you study large conformational changes, the traditional all-atom models usually are not very useful because they take too much CPU, and and then you have to consider very large, very short time scales, meaning very large speeds and so on. So we do a coarse-grained model in which amino acids are represented by balls, and they are like they are 3.8 angstroms apart. But that's a model of a polymer, and when you introduce attraction between certain beads, then it may acquire a shape of a given protein if you select your couplings correctly, okay? And there are various ways to do it, but the one we use it is like this, that we read in the native structure of the protein with all its atoms, and we assign van der Waals spheres to the atoms, and then each amino acid is then represented by a cluster of grapes, and then if these clusters overlap, then we say there is a native contact. And if that happens, then we assign some attractive potential, and the simplest choice is take Lennard-Jones potential with amplitude epsilon, which we calibrated it to be of order 800 Kelvin and 1.6 kilocalories per mole. And the calibration, we did the calibration by comparing the theoretical data on stretching to experimental data on stretching, okay? And so these are the experimental results, these are the theoretical results. We also did extrapolations to speeds of pooling that we actually used in experiments. And this is based on like 38 proteins, and the best fit of the simple model is that the effective epsilon is of order 100, the unit of force, epsilon over angstrom, is like 110 piconewtons, but say plus minus 30 is an error bar. All right, now I want to discuss, it's stubborn. Oh, sorry, so, and as I told you, we have a database in which all these calculations are deposited, and you can learn about it. And I told you that the two typical, this characteristic, the reason for emergence of force peaks is usually due to shearing between two beta strands, and the longer they are, the bigger the shearing. But we have also discovered things like topological mechanical clamps. Mechanical clamp is the motif responsible for emergence of big force. And this we called it cysteine slip knot, which involves dragging of a loop through a slip knot loop through a cysteine ring, which is like a circle, and then a big force is involved in doing this. So, and it shows us a force peak like here, and the characteristic force could be could be going to like 1,000 piconewtons. That is like 5 times stronger than titin, or even perhaps stronger. And now, so let's now discuss virus capsids. So there are millions and millions of viruses, but only of order 5,000 have been described in detail. And less than 10 were studied by manipulation by AFM and the similar methods. So far, and so they were discovered in 1998 in Holland, and the first object that was, first virus that was discovered was tobacco mosaic virus. And it's not spherical in shape or quasi-spherical in shape, rather it's elongated or helical. However, there are all kinds of viruses, and those which were studied mechanically so far were almost all quasi-spherical. So in particular, this one fellow called CCMV, which stands for cowpea chlorotic mottle virus, and chlorotic means this color that's, that leaves get when the virus sits on the plant. And cowpea is another name for black-eyed pea. So this is the pea, and it was domesticated in West Africa 5,000 years ago. Okay, so another virus that was fairly well studied was polio. And this is spherical. And so was the CCMV, to which I'll come back again. That would be my workhorse. I mean, example I studied the most. And it is a capsule which the outer radius of which is like 16 nanometers or 160 angstroms. The inner radius is like 9.9 nanometers, and it consists of 240 proteins, and and the total content of amino acids is like 15,060. So it's a big object. Okay. So then it's an ideal object for our model, in which we use coarse grading, and therefore we can study either thousands of proteins, or we can study big objects such as this one, as a capsid. So basically, the capsid is a shell, and it looks like a sphere. And it actually, when it comes to nerve cell in this case, then it could be captured by a receptor which sits on the membrane of this nerve cell, and then it could be absorbed through a process called endocytosis. And it looks like the life cycle looks something like this. So when it is absorbed through endocytosis, then it takes over the machinery of ribosomes, which produce proteins and apparently also produce RNA. At least that's my understanding. So pretty soon the cell produces thousands of new copies of these viruses, and what it produces is proteins which are involved in making the shell, but also in making RNA which sits inside of that shell. And the shell contains RNA, actually in this case I believe four single strands RNA. So people in Oxford, for instance, try to make models for self-assembly around RNA, but it's an interesting process, but these are very, very coarse-grained approaches in which you simulate, you collect plaquettes or something and not molecules, so that would be interesting to study. Now there is a database called ViperDB, and it sits at Scripps, and there's several structures, there are hundreds of structures there, not all of them complete, but basically this is like a offshot of protein data bank which is focused on viruses, and it gives you the coordinates of the atoms in and RNA and so on, or if it is an RNA virus, there are viruses which contain DNA and so on. But anyway, this would be the input to my studies. So I need the coordinates of these atoms in the native state of the shell. Now, this quasi-spherical, yes?
Do people study crystallographic groups of
Yes.
viruses we just showed?
Mm.
It's well understood?
Well, okay. I'm actually coming to the subject in a way. So for instance, so this capsid is quasi-spherical, but it's not exactly a sphere. Okay, atom sits in, okay, they vibrate around some well-selected position, and so it was tempting to model structures by doing, polygonalization of a sphere, and there are many ways of doing it. I mean, infinite number of ways to put polygons on a sphere. but you can enumerate them. And in particular, those which are interesting for this quasi-spherical capsids, have pentavalent or pentameric units and hexavalent or hexameric units. So let's depict it like this. This is the pentameric unit, and this is hexameric unit. And the question, and this is one of many types, and this type is described by a T number equal to 7. Well, T number, I put subscripts here after Caspar and Klug simply because I need to distinguish it from temperature, which is another parameter, in the model, but usually it's just called a T number. And it's defined in this way. These are two integers, h and k. And they have a meaning, and this meaning is, for instance, that in order to go from one pentamer to another, you have to go across two hexamers in between, okay? So that stands for k of 2, okay? But you don't go along the straight line from here to there in the structure, okay? You have to do one sideways motion and then two steps, and that's means h of 1, okay? And this, if you add this, then it's 7, okay, and that is a well-defined structure.
Why does this particular combination has a meaning?
Well, this is an example of a triangularization.
No, I'm saying this particular combination of H and K, is it geometrically inspired, or?
Yeah, it's pure geometry so far. I mean, but then when you have a crystallographic data, you want to interpret them in terms of what particular virus geometry would fit it the best in terms of symmetries that are involved.
What I was asking rather, if you have two H and K numbers, right?
Mm-hmm.
Why do you combine then this H squared plus K squared plus HK, not something else?
Okay, because this number, it tells you how many pentamers and hexamers you have. Okay, so for instance, this T number tells you that you have 60 times this T, this is the number of proteins, subunits of proteins, that tells you how many proteins are involved. And then you know that each type, this is T of 7, T of 4, T of 3, so T of 1 is one in which all you have is pentamers. Okay? But then you keep adding hexamers and the structures become more and more complicated. And this, we know that always you have 12 pentamers, but then if you know the T number, you know that you have 10 times T minus 1 hexamers, and things like that. So the T number determines everything that you need to know. And this is Caspar, I believe he's an American. And this is Klug, he got Nobel Prize in '82, not specifically for this work, but for his general contributions to chemistry. And so they proposed this geometrical model as a model for structures of virus capsids. And so I'll be interested in all of them. And in particular, so let me now go to, in particular, in CCMV. So as I mentioned, CCMV was particularly well studied. So this is an X-ray image of the virus. And of course, this is a molecular structure. This is not a sphere with triangles or something. So it takes big effort to map this to particular geometry. Now, this is an AFM imaging picture. So you can study the capsid with RNA inside or without. Okay, both were studied. Okay, in my model, I need to start somewhere, so I do it first empty. But the hope is that future studies we can study capsids with RNA, but that we have to develop it better. And it turns out that the empty is actually bigger than the empty is bigger than full. So for instance, the full is like 18 nanometers across, whereas empty is like 22 nanometers across, which means that RNA does play a dynamical role in keeping these proteins together. But it is stable without RNA as well. So that's the first thing to consider then. And it was studied at UCLA and in Amsterdam, and there's a long-standing collaboration between this university, Free University in Amsterdam and UCLA. And this is some names here. I'll list more of them later, like Guy Wout, if I pronounce it right, is one of the leaders there. Bruinsma is a theoretician. Now, so here what happens is that you do nanoindentation, and initially you have an elastic response. Okay, that's Hooke's law. That's Hooke's law. Okay, but then at some point there's a collapse, and suddenly this virus, gets flattened. Okay. And then you can squeeze it further, and of course the force would be growing and growing because of hard cores, you know, because of the excluded volume.
Because this curve goes up to like 30 or 40, and you just said that it's 20 nanometer across.
No, this is this is something else. This Z, what is measured here, is the indentation. So there's one plane which is flat,
Yeah.
okay. Then there's a capsid, and then this is the distance you go in from the top.
Okay, but the zero means that
You're
they
touching
both
it.
touch.
Yes.
But this means that you have moved this upper plane by more than the diameter, so they should, I don't know, they should touch the plane.
Oh, I see. Well, yes. No, but it. Yeah, it also takes this Z takes into account the deflection of the lever This takes into account the bending of the lever, and then you have to remove that when providing information about spring constant and so on. But then when you reverse, you keep squeezing, and then you revert the motion of the plate. So one plate you can do both or one. I mean, depending on your mood. But then this is quite collapsed, and it takes minutes for this to recover back to the native state. So when you move the plates back, then there would be no force felt by this because it keeps being squeezed, and But after a long time, it would recover. And now, it says that if you take, say, a spring constant here, then if it is full, it's stronger. I mean, the bigger elastic constant, 0.2. If it is empty, it's like 0.15 newtons over meter. That's the unit of the spring constant.
Can I have a question for the previous one? That minimum and maximum in the indentation curve, when you see the drop of the force, is that the buckling of the sphere? I mean,
There could be,
yeah.
Mechanically. Well, I mean, this is the experimental result, and now you want to interpret it, okay?
Right.
And in our model, we do see some buckling for CCNV. Okay. Now, so I'll focus first on two virus capsids, CCNV, cowpea chlorotic mottle virus, and there's a related virus, CPMV, which is just cowpea without anything chlorotic about it, a mosaic virus. This has this T3 structure, so the T number is 3, okay? Now this is PT3, and this means pseudo T3. And there are exceptions. I mean, not all viruses are covered by Kluck and Kasper classifications. I don't know, many of you, but there are which are outside of it. And but one example is that the proteins involved need not to be identical. There could be two different proteins, okay, and that is. But still, it may look like a T3 capsid, so it's called PT3. Or one protein is involved in the one unit, and then another is involved somewhere else. I mean, it's bent. It's also pseudo. Or the number of proteins actually bigger than what you would expect for T3, but they effectively one act like an extension of another. So anyway, right. So this is the CCMV is modeled by this. Figure. So it's a truncated icosahedron. That is, of course, it has 20 walls. And it has 28,620 C-alpha atoms. That is that many amino acids. At least the position of those that we know the positions of. Because there are some which are the position is not determined. And if I do this procedure of evaluating contacts, that would have like 62,000 contacts, interactions between amino acids. Now CPMV is similar in size. It is that both contain like 300,000 heavy atoms, but it has not 28,000 but like 33,000 C-alpha atoms, and it has a much bigger number of contacts of 90,000 something. Okay. But both contains this contains of 180 sequential identical chains, whereas this has two different chains. Okay, now, so we take our model, and apply it to CCMV and CPMV, and we want to see are there any differences between them, okay, because they're sort of similar in geometry, similar shells geometrically. Would that be any different mechanically? Only CCMV was studied experimentally, CPMV was not yet. And this work was published, part of it was published and was done with Mark Robbins of Johns Hopkins, with whom I collaborate for some 25 years now. And another big manuscript is being completed. But now let's look at distribution of the C alpha atoms. You know, if you sit at the center of the Earth and go in the radial direction and see the distribution of mass, then you see it's shell-like. So obviously it's not a thin sphere. The sphere has a width, and the width is of order 20 angstroms if you measure 2 standard deviations as a characteristic measure of the width. Of course, the system have been studied theoretically as well, and also it started all at UCLA. In particular, Gibbons and Klug, another Klug, not related to Aaron Klug, who got the Nobel Prize, but he's a professor in engineering at UCLA. And they proposed to use shell model, a continuum non-uniform elastic shell model to describe what's happening. And we find that in terms of the force versus indentation curves, you get behavior which is consistent with experimental behavior. However, but the point of my talk would be that there are many molecular level effects that describe the behavior of the capsid, and if you look at the strain field, that would be very different than what the shell model would predict. Okay, and furthermore, this molecular model that we use is coarse-grained, but it is molecular, and so you can distinguish between contacts between the proteins and within proteins and see their role. And furthermore, if you have a shell model, then you can do only T equals 0 calculation. T now means temperature, but in the molecular model, you can study the effect of temperature, and have thermal fluctuations. So the point. Yeah, so this is model of a particular, so you adjust the thickness to the capsid that you are studying. This is for CCMV, this picture.
Calculate the thickness of shell from the histogram, right?
Right, so from the distribution taking the width.
Does the histogram include Jacobian R squared? Because you will get slightly different.
No.
Well, as you increase the radius, the number of things should be proportional to R squared. So does your histogram divide?
No, I understand what we're saying, but we didn't think about it. We just look.
It could be quite a fact because you have like 10%.
You might be right. I guess we didn't think about it. But still, it's the order of magnitude should be, what we. So now what happens? So this is shown here. So basically, we prefer to squeeze from both sides, and we start by using flat planes, okay? And so the S will stand for the separation between the planes, okay? And then we'll consider curved planes as well, but we start with flat because that's the first, simplest case. And then this is the native conformation. I mean, this is a very crude representation of 28,000 atoms. No, the 28,000 atoms are shown here, but you don't see much. And these colors mean nothing, they just come from PDB. And so it's hard, but basically you see that on squeezing, this thing gets flattened. Okay, sounds reasonable, but this is not what you get in the shell model. Because in the shell model, if you think of a football or a European football, or a beach ball, and you squeeze it from the top, then it would bulge out at the equator, okay? Whereas here, something that shifts up and down, vertical, around the equator, as I'll demonstrate later, but nothing could go out. So that's the first difference, okay?
I can't see it in this one, but sometimes what happens, think of a ping pong ball, the dimples also,
Yes.
it increases the
Something like that. I think it will do well. Tomorrow, I'm showing the picture for CCMV, whereas what happens in other cups, and it could be this. I mean, you have to analyze it in details. this is showing what the top atoms, for top 2000 atoms are doing. This is the native state. So the separation is equal to the diameter here. But then, it just keeps rolling and it just flattens it, okay? Now, and this is a more schematic representation when, thousands of our atoms are represented by single dot. And this is the pancake conformation, which corresponds to going beyond the yield point and correspond to the collapse. And this is the further collapse. Now, you can do it, of course, at various speeds, and we happen, if you take typical speed, then our model is just 5 times slower. It's 5 times faster, that is. We can do it really at experimental speeds, but there's only a difference by the factor of 5, so it's not that bad compared to all atom simulations, which would differ by 7 orders of magnitude and such.
What do colors represent? You mentioned, but I missed. What do colors represent here?
I said nothing.
Oh, nothing.
No, this is some automatic assignment of color, but but of course, if you want to, there would be better ways to represent it by focusing on various motifs and structures, but it's not done here. Just too many to worry about it. And 20,000.
You compose it from these little.
So I read in the full structure.
Yeah.
Right. So. And
So they're the subunits, probably, of the individual connected chains.
No. You see, these are the regions. You see, because this is this is the south pole.
It may be the pole where the DNA is ejected, or the RNA is ejected when it sits on
No,
the cell.
no.
Because these things have orientation.
No, I know, but I'm pretty sure that
is not
this is that the PDB doesn't know about it. You have to do your own intellectual work to make proper colors here. So let me show you the force versus separation curve that we get in the model. What happened? This is the curve I will focus on. So first, you have a linear response, okay, with fluctuations because we have thermal fluctuations. And this is reversible because if you stop here and then withdraw your plate, then the return path would be one in purple, okay? So within fluctuations, reversible. However, if you go to this yield point, then it stops to be reversible on the similar time scale at least. And this is fully reversible. So this is the stage here when it's flattened to this pancake. So this is the curve which goes up when you indent, and this is when you withdraw. Okay? And this force that I'm plotting is actually an average between the force seen by the top plate and the bottom plate. So the top plate is in the red color, and the lower plate is in green, and what I'm showing is the average. But of course, this is a living object. I mean, the computer. Object that lives in the computer. So that is, if you take another trajectory, that if you change your random seed number, then the trajectory is somewhat different in general, but they start about the same. See that there are 3 trajectories corresponding to the speed I was mentioning, and they are roughly the same. But then the yield point may depend on the trajectory. Yes, you have?
Yeah, I'm just wondering what happens after you go through the minimum. The separation, you keep the force.
No, I keep the distance, the separation keeps decreasing at a steady rate. Okay?
Right.
But then I may stop at some point, like here at 0 there. And then I keep evolving it, but now the plates are moving away. I'm from the capsule.
Do these hysteresis curves depend on the speed at which
Right.
they do?
Yeah, so that's the thing. So, okay, so now I'm showing 3 trajectories for this original velocity. So initially they are, they're equivalent, but then they split around the yield point. So even for the same velocity, they make a yield point differing somewhat from the trajectory to the trajectory. But then, if you increase the speed of the wall motion by the factor of 2, then you can extend the elastic regime. Okay.
The wall is not connected in any way to the virus, right?
No, it's
just a moving plate. It's not like in AFM that you actually attached to a certain amino acid. Here you are just moving it.
No. You can you attach to certain amino acid when you stretch proteins. But
yes,
here,
but I'm asking here.
no, you have just a plate and interact through repulsive interaction with the atom.
Yes, but when you move the wall away, there is a possibility that it would not be
attached
connected.
to
Or
Oh, I see.
attached to
No.
virus, yes?
It's only because we model the interactions of the capsid with the wall by repulsive interaction. If we change the model so that it has some attraction, then of course you might try to pull out.
Yeah, but
Maybe
that's
it's
okay.
a
I'm just
So it's
trying
a
to
finite
understand.
range repulsion, it's
Right.
not actual contact.
correct. But of course we can consider something which has attraction and that would behave somewhat differently. But you have to start first things first, I mean, what if it is purely repulsive? But if you make it move slower, then this yield point would be, you arrive at it earlier. Okay, so that you see that's a characteristic of viscoelastic behavior. That it behaves like solid, long, for bigger, longer time if you do it fast, and then the speed of approach of the place.
Fast means
Twice as fast than this speed here, and slow means reduced by the factor of 2.
okay.
Compared to this one, 50 millimeters per second. So clearly we have some thermal activation processes, and this something that cannot be captured by shell models. Now let's look at the values. So the characteristic force, the yield force, I mean, we can order, since it depends on the trajectory and so on, we can just speak about orders of magnitude. But if you do the calibration that I mentioned at the beginning by comparing, stretching data to experiment, then this force peak should be of order 600 piconewtons, which, and experimentally it's of order, it's about the same, okay? So that's good news. However, if you look at the spring elastic constant, then it's smaller by a factor of 3 roughly compared to the experiment. But we understand it because, you see, in real life, amino acids are represented by many atoms, right? Here it's represented by a single ball, so the space is more empty around it, okay? So it's more pliable. So that's just the feature of the model. All right, now, of course, the results in principle may depend on how you squeeze, If you squeeze it one way, this way, then this is the white curve here. But if you squeeze along this direction, then you get something, which within error bars and thermal motion seems to be similar to what you got for the other direction. However, that could be a feature only for CCMV. Maybe for other viruses, it's different. It has to be investigated.
Squeezing direction means you're choosing something not in the symmetry group.
Yeah, this is the biggest axis, the largest symmetry axis, I
Right,
believe.
so you have the largest diameter, the shortest diameter, and things, right?
Yes, yeah. But so there is some sensitivity perhaps, but not much. Now let's study, this is the curve I was showing. Now you can study it without any thermal fluctuations, and what you get is that the peak, which at a different location and much higher. This is meant to be roughly room temperature situation. Now these are other temperatures, so this is higher temperature, these two are higher, and this is lower, and this is the lowest of them all. It has no thermal fluctuation of all. So you see that peak height and depends on the temperature, but the elastic constant is not very sensitive to it. I mean, it's weakly sensitive. There is some dependence, but it's about the same slope. Now you can also study what happens if you do it differently, either by using this thing called force field apparatus when you have two cylinders that squeeze things, that's done in Santa Barbara. Or you can attach a colloidal particle to the AFM setup, and then this would be a broad surface. And you can control the curvature of that. So I understand that typically the curvature used in these studies is like 30 nanometers. So this is a plot which shows what happens if you go from flat to curved. So this is flat, okay? And now the curved is And this is curved at the same temperature. The white, this white one is curved. So you see that it has a different, it's shifted force peak, I mean the value of max or Fc, whereas the initial elasticity is comparable. Okay, but there is some sensitivity to that.
What's the scale of the native thing? The curvature is about, also about the...
Oh, the radius of the capsid is, for CCMV is 100.
100.
Okay.
How are the capsids attached to the bottom plate?
Oh, this is some, I don't know. I mean, this is the biotin streptavidin something. Something well known, but I don't really know. I want to visit a lab which does it, but I didn't do it yet. Okay, now let's look at another. So this is CCMV, and I mentioned that I can study it microscopically. I can see what happens to the contacts, and I see that at the collapse point here, half of the contacts break down. That is, they exceed certain characteristic distance. The separation between amino acids is too big. And interestingly, at least in CCMV, 92 or 3% of them are interproteinic couplings, because there are contacts within proteins and between proteins, which act like hinges or something. And at the collapse point, it's the interproteinic contacts which dominate. The breakage of them dominates. All right, now let's look at CPMV, the other virus capsid. And geometrically, it looks similar, about the same radius, about the same width, okay? And yet, what you observe is the force, which is like 3 times as big. And the elastic constant is significantly bigger as well, okay? So the question is, what is it due to? And that's what I'll be focused on now. But the first thing we proposed was that, okay, if you look at the average number of contacts, that an amino acid is involved in, then in CCMV it's like 6.4, including the contacts along the backbone, which is the tethering contacts. Whereas if you take CPMV, it's like 7.4. Okay, so this is closer to the FCC. In the FCC, it goes in the FCC direction, in which you have 12 neighbors, right? So it's more like solid. So let me now go to other viruses, and this is now what we are working on. I mean, we did a survey of 35 capsids. 4 of them were studied experimentally, and HK must stand for Hong Kong, but it's a virus called HK97. So this is the group, Dutch and Purdue group, and Scripps, I mean, various places involved. But anyway, so this is a small virus, MVM, and this is of T1 type, and it has a simple structure. So what I'm showing here is this flat curved squeezing, and the yellow thing is the curved squeezing. I mean, if the tip is curved, okay? And this shows T equals 0 situation for comparison. At T equals 0, there is no force peak. Now, this is NV. And I keep forgetting what these acronyms mean now, but it has a shoulder. This one there are 2 trajectories. Sorry, this 2 is one is higher, the other is lower, so we take average of the characteristic force. But also there's some shoulder here, so there's some structure. But if you take HBV, then you see this pattern is very complicated, it's very wavy, okay? And this is of the T4 kind, so many hexameric units. And HK97 is it's still different, it's like a plateau, with the force peak and so on. Now, if you compare the characteristic forces between the experiment and the theory, using our calibration obtained for proteins, then you get a decent agreement considering the simplicity of the model. But if you take stiffness constant, there's a uniform shift away from the experimental data for the reasons that I just specified. The model is emptier, like an empty planetary systems. So it's encouraging that you could, perhaps compare various proteins like this. So this is the latest work. This is on the 35 viruses. And these are the values of the characteristic forces, and elastic constants are shown against the average radius. Okay. And so you see that many of the T3 and PT3 viruses have, have closer, has similar values of the radius. Not every capsid, but many of them are. And this radius is 132 angstroms. So it's not the outer radius. It's the average radius. And CCMV is here, and CPMV is there. So you see, accidentally, when considering CCMV and CPMV, we really cross the range of values in the elastic constants. But what you see is that there's a big, it differs by order or by the factor of 30, 20, depending on whether you consider the elastic constant or the force, but it's a big range of values, big range of behaviors, and so on. So there are some names here. NBV stands for nude Aurelia, I guess, beta virus, in fact, spine emperor moths. So that's the caterpillar stage of the moth, as taken from Wikipedia, not by me. I wish I went to Ethiopia to do it myself. Sorry? No, they feed on pines. And moth this is the larva caterpillar of the moth. Okay. Did you get hungry? So the reason it's singled out here is that this is, I'm sorry, where is this fellow NBV? Here, because you see it has one of the biggest values of the elastic constants, and also the reason why I wrote something about it, that this is the biggest capsid we have studied. It involves 135,780 atoms, and one job runs for like 5 weeks on our workstation.
How many molecules does that mean?
Say it again?
You said that's the number of atoms.
That's the number of C-alpha atoms. That is the number of amino acids.
How many molecules are in this?
Yeah, that's the number of molecules.
It is the same.
Yes, it's the same. Okay. Whereas the number of all atoms would be this times of order 10. Okay. And the width of this is 43 angstroms. Now, the largest one in terms of geometrical size is this HK97, but it actually has a fewer number of atoms. Okay, and the width is smaller. Now, the smallest capsid that we studied is SPMV, which stands for Satellite Panicum Mosaic Virus. So it's a plant virus. And so this is this one. It contains only 8,460 molecules. And the width is like 13 angstroms. And this is simian virus 40, which is also pretty big in terms of the second biggest in terms of the number of molecules. It's 123,420. So you see, this is the range of values, and the question is, what does it depend on? This is one common value of the radius for this T3 and PT3 viruses, and these are 2 other common values, like for T1 viruses here, and another set of T1. So this is 73 angstroms and this is 109. And the other are scattered, and in some cases, like T2 virus, we have only one example of it. The only structure that we could find.
Do you have explanation for these preferred sizes? Why they concentrate in these groups?
Well, they are sort of similar. No, this is a biology thing. I
So
don't.
they are related, those viruses, in one range?
Well, I see you'd have to know the evolutionary story to see what came from what. I mean, I wouldn't bet that this is specifically evolutionary related, this to that, okay? I don't know. But they happen to prefer, maybe it depends on the sort of number of atoms involved and interactions involved.
So
there must be something common.
From the mechanical point of view, you see no reason, yes, why this particular size.
From mechanical point of view, I see no reason. However, there must be a reason. I mean, similar number of RNAs. I don't know.
But you don't know that.
I don't know. For one thing, we just noticed it recently, so so we have to first notice it and then study it further, because when doing calculations, I thought the values of the radio were all scattered, okay? Because, okay, different numbers, but when you actually make a plot, then you're surprised to see that many of them are similar, but that came much later, you see. So okay, so now this is the value of the forces, and this is the elastic constant. Now, so let's say elastic constant. So if you take this PT3 capsid, it's something like a trend, whether they have same value of the radius or not. And now if you have T1, then the same radius, if there is a trend, which is hard to see, then there are two separate trends. Okay, for one radius would be this, and this would be for another. Whereas this agrees, sort of goes into one curve. So it's all kind of values, and it's hard to make sense of it, really, for initially.
So you normalize by the radius of the overall structure, not of the individual subcategories.
Okay, just wait. So first I'm showing you the raw data for elastic constant and force. But now I just showed the values. Oh, sorry, I forgot to say the most important thing. I'm plotting it against the average coordination number. Okay. Coordination number, that is how many contacts a given molecule is involved in. So I'm trying to understand it, you see, encouraged by the results on CCMV and CPMV, where the difference in coordination number appeared to play a big role. We think, okay, let's try it to other viruses, maybe they explain me something. Okay, so they explain something, that there's some trend, that the bigger Z, then you tend to get stronger capsid. Okay, but it's hard to make one simple trend, and even if you focus on objects which have roughly the same radius, then these, they maybe one makes sense, but these are two different trends. Okay.
I'm just confused. I mean, I thought things were made of hexagons and pentagons.
Right.
So why is the coordination number shifted a little bit to the right?
No, but these are all, green objects have same number of hexagons and pentagons, because these are T1, same T number.
Right.
So the only thing, what differentiates between this could be coordination number, could be the width, could be the radius, and maybe something else that we want to find out. What is that thing that governs the strength?
I'm just trying to get at what the coordination number is.
The average number of contacts and a molecule is involved in. So typically it's like 6, 7, something like that. Which means, see, if you have a chain, then there are two tethering contacts to the left and to the right along the chain, but also the attractive interactions to, say, 5 or 6 other amino acids, and the total number of that is Z. And I am now plotting this against average Z, encouraged by what we knew about 2 viruses. Now I'm trying to apply it to 35. And it's not that simple, you see that Z does play a role, but it's but it's vague, okay? But now we got something just actually 3 days ago, that if you plot the Young modulus, then you start to make sense in it, okay? So elastic constant depends on the radius, depends on the width, and so on. Okay, so let's extract this geometrical information by introducing Young constant, so that E is defined as k times r divided by h squared, where h is the width and r is the average radius, but in this case we say average radius. And so this, you can think of it as just obtain. It was first, I've seen it first in a paper by Gibbon and Clock, but you can think of it as a dimensional consideration. Find a quantity which on dimensional reasons should not depend on the geometrical parameters. And let's plot that, okay? So now this thing is, all these viruses of 6 classes that we have, not all of them are, some of them are represented in bigger numbers than the others, but then you see there's a trend which seems to be parabolic when you do study deviations away from 6, which is the threshold of stability. So if you have a z, average z close to 6, then you expect a small value of Young modulus. And you see a trend. And now, but there are outliers. And the question is how to understand why do you have outliers. And the recent realization was that these 4 viruses, STNV, IBDV, and whatever, and the other 2, actually cannot be considered in thin shells. You see that if you take a threshold, that the ratio of width to radius is bigger than something like 0.24, and the threshold is like 0.24, then these 4 above this threshold. Okay. So whereas this form, so it looks that at least when you can use this thin shell approximation, that there is a fairly well-defined dependence on the average coordination number for Young modulus. So now you can explain back these other results.
When you
Sorry?
The shell is more surface-like than solid.
Right. That's what it means, that if it is thin compared to the radius, but of course thin may be 40 angstroms, for instance, and yet still the object could be, thin shell.
When you calculate your coordination number, you said you just basically count number of neighbors.
Right, in the native state.
But do you include the relative strengths of interaction?
No.
Just cut
You do
off.
not, or you do?
No, I do not.
It cuts off.
But
No.
maybe that would be more appropriate.
No, maybe, but the model actually has the same, the potential wells in this simple model that I'm using, they have the same depth. So in the native state, the strengths are identical, well, or close to.
But the further the molecule, the thing is, the weaker it interacts, right? I mean, the true neighbors interact with their strengths, but next.
No,
next to neighbors interact
but the
less.
range, as I said, the contacts are determined by representing amino acids by a cluster of grapes, okay? And the size of the grape is 8 angstroms, 9, I mean, nothing bigger than that. So if the object has to be close by in space, okay, to make a contact, and it never exceeds 14 angstroms. So the contacts that are involved cannot be bigger than 14 angstroms, 14.2 or something like that. Okay.
So, Mark, I think one of the confusions I have is that because it's a fairly thick shell, the
Mhm.
coordination isn't completely determined by the plane. It's really, it's actually, Keith just said it's really a thick thing, so it's a 3-dimensional coordination.
Right.
Yeah, that's why I was
Right.
6 as opposed to 12.
Right. But we always say that if you can isolate objects which can be considered thin shell objects, all of them are 3-dimensional, of course, then you get this dependence on the average coordination number, which we have to understand better, but I didn't know about it 3 days ago. All right, now let's go back to these 2 viruses, to CCMV, okay? And let's try to study it in more details. And so we want to study strain field, because I told you that if you plot force versus separation, that it's similar. But now let's look at the strain. So so what you do here is that you sit at the equator and set the radial directions initially in the native state, and look at look at the location of an atom there. Okay, and then on squeezing by some distance, okay, in this case, if you squeeze it to 202 angstroms from 250 or something initially, then this point would move down. Okay, so you can plot it versus the distance away from the equatorial plane. So here, positive H means shell points which are above the equatorial plane, and negative H means shell points below the equatorial plane. Now, but what is the quantity plotted here? Well, it's the radial strain. Okay, so you define it in the following way: that you notice the native location of an atom, and then you note its displacement dr, and you make a dot product, of the r and dr. And then you normalize it by r² for the fun of it, so that it is dimensionless. And this is what we get in our model. So what it says that in the radial region, the strain field, the radial displacement is essentially 0. Yeah, okay, but each point is a single C alpha atom. So you have 28,000 points here. Okay. Now we want to compare it to the continuum mechanics result, and we got the data from Klug. And so now, of course, there are no atoms there, but you can take the shell and discretize it and select certain atoms, so to speak, and see where they went, in his experiment. And this is what, and do equivalent thing. And you see it's very different in the region, in this region, because you see it's like 15% bulging out effect, which is very different than what you get in the molecular model. Now there's some asymmetry here because we squeeze from two sides and they squeeze from one, but you see that there are some qualitative differences. We also see some backlink and some in rotation, but we need to study this backlink better. Now another thing, it's getting late, so I don't know how much, maybe I should quit soon, but now you can also look at this, this is the plot in which you know the location of an atom, and plotted vertical location of an atom, and plotted versus the native location, vertical location, h of s versus h. So you see these are all points that are involved here, and we can do it for specific regions, pentamers, hexamers, and so on, but you see there's a shift, it's sort of tilting here, so that means deformation. Sorry, there's a shift, but also there's a deformation at the equator. And when you compare it to the shell model, it would be different. And one way to compare it is, for instance, this plot, is that here I'm focusing on if I take the atomic model or molecular model or shell model, then I look at points which are on the surface, outer surface, and on the inner surface, okay? So the outer points are shown in red and the inner are shown in blue. Okay, and then I note what happened to this point. So in the molecular model, even though there is no radial bulging out, the displacement, there are vertical shifts, you see, around the equator. Whereas there's none of this in the shell model, which only works radially out. And then the pattern at the equators, at the polar regions is distinct as well, but let me not dwell on it because it's late. So this is so we have this C-alpha based description of proteins that we use to make surveys of proteins, mechanostability of proteins, but also now to study mechanostability of virus capsids. Right now, these virus capsids are empty. We'd love to extend it to capsids which contain DNA or RNA or DNA. And we studied flat surfaces and curved surfaces, and we see that first you observe elastic region, which is then followed by reversible behavior, which is activated thermally, and it goes into a sandwich shape. And we see that the force of resistance, that even though the plot of force versus separation agrees qualitatively with the shell model, then there are many disagreements when you start to study the strain field. And in addition, there are the thermal effects and so on. And finally, the latest finding is that many of these viruses, capsid viruses that we studied, when they can be considered thin, then there is a fairly well-defined dependence on the average coordination number. Thank you very much.
Indeed, Marek. I was just thinking, what could be the medical implication of your work, in the sense that you have all these contact points, and then this determines the stiffness.
It makes me sick.
And then it would be how maybe if you could study, for example, which bonds you have to destroy in order to weaken the structure. So then you could target with some drug these points and maybe disrupt the capsid. So is this possible to do?
It is possible, but it's a tedious work, and so we did it really only only for CCNV. And there, as I said, the collapse is governed by the interproteinic capsids, contact, that is.
Contact, yes.
Okay, but whether it's a general rule or not remains to be seen. Okay, and furthermore, there's
contacts which are more important than others, because you treat them all on average. But
right.
For example, if you've got some, maybe it gets destroyed and others not. So are there indications of this kind of
Uh,
behavior, or
Well, some. I mean, we see where when the process starts, when the collapse starts, what bonds are ruptured. You know, we can monitor it over time. And so maybe these bonds which are ruptured the first could be those which could be targeted. Yes. So we have a plot, but then I have a plot how it looks, you know, as a function of separation. But then I would have to go back to the list of these contacts to tell you what are these.
Yeah, my question is related really. What is the there has to be some kind of medical motivation behind this sort of study.
All right.
It is of course interesting to people that are
Right, so that's what I thought. And maybe there will be. So I thought that maybe this is related to, you know, lots of work of encapsulation process, you know, that you want to put things into, you know, that the capsid ideally could house something that could be delivered, right? So maybe you can do it mechanically or something, I don't know. But I thought, but you know, there are so many people, like 30 people work on viruses at UCLA, and but they never mention what is it for, okay? But a year ago in Santa Barbara, I met one of these big guys in the business, Nobler, who is a physicist, and I asked him, is it secret, is it some patent, or what? Why you don't ever mention, you know, that, you know, what is it for? And he says, we just do it for the fun of it. And, but for me, though, is a first step in studying larger systems, because the objects are well defined, have good symmetry, they're fairly simple. So if you master this step, then you can go and study even bigger systems with this model. Okay, so from theoretical point of view, this makes a lot of sense to me. Whereas experimentally, I mean, I believe him that it's for the fun of it, but maybe still he doesn't say the full truth.
Roman.
My question is, has anyone or does anyone know anything about the amino acid content in a general sense for different kinds of capsids? That is, whereas you're looking at things at the level of C alpha, maybe you have 2 or 3 kinds of C alpha. Those that might interact through salt bridges, those that might interact polarly,
Right.
and then you might have some rationale as to why you see this difference. Has anyone ever looked at that?
Not as far as I know, but no, it's a good question. But as I said, first things first. You know, first you do the simplest model, and then you can try to identify the relevant region and maybe model it differently to see the difference. Yes, it's a good question.
I think that capsids are rather designed to resist to internal pressure, right? The DNA is packed into capsid, and it must keep DNA very, very compact. As you said, if you take capsid which is inside like 7 nanometers, this is much less than persistent length of DNA. So I wonder whether you can use your simulating tools to see what the pressure capsid can resist.
I understand the question. So, well, it's hard, you know, in this model, the solvent is only implicit, right? So in order to study things like pressure, you need to have more properties of the fluid, you know. But this high pressure virus capsids are not those, okay? Because I was focused on small capsids, which are, they typically contain short pieces of RNA, 50 nuclear base pairs long or something like that. Whereas those which have...
So those are RNA viruses, not
yes.
DNA? Aha, okay.
Or at least most of them. But if the DNA in a short segment, nothing big. However, virus capsids that hold, you know, they come with large pressure, and they typically also house DNA, they have a different structure, because in addition to this inner, they have outer shell, you know, which is the name, but glucoproteins, you know, something much harder. Okay, so you have this inner shell and then outer shell, which is made of glucoproteins, and then that is meant to be called good pressures, but also These DNA containing viruses often do not look spherical at all. They look like spaceships and other, objects that can threaten your life because they have some, a spear and something, but that's due to this outer structure layer. So that would be, and these are bigger in radius, at least by the factor of 2 or 3. So these are still larger scale objects.
Excuse me, have similar studies been done in, say, C60, like buckyballs? I mean, much smaller systems.
They must have been, but I'm not, yes, I am, theoretically,
What's one of your Frank Casper
no,
structures?
it must be, well, I didn't see AFM studies of C60, but they could be small. I mean, that could be too small for this, you see.
I thought that was one of the structures. It's one of the Frank Casper structures.
Well, no, but it's a different thing to study theoretically and a different thing to study experimentally.
You studied it as one of the...
Right, but the problem is that if you, AFM is, has its limitations, okay? And one of them is that you cannot study too short distances because there are apparatus-related effects, image charges and such, and you don't know what is the signal and what is the apparatus, okay? And I suspect that if you take a buckyball, that would be too small an object to study directly this way. So in order to do it indirectly, I guess you have to put in some padding. So I don't know whether it was done or not, but it's a good thing to inquire
I
about.
was going to follow on that. So I didn't realize it literally about the bulk, but you could crystallize it and study it more in bulk, the same.
So experimentally it was studied through crystallography, of course. But I mean, mechanically, I'm not aware of that, but maybe it was.
Studied the mechanical property.
So I have two comments.
You
and one
educate me
question.
about something.
Well, so this is my department, so I listen to these students all the time talking about viruses. Some of it has come in. So one of the latest things that they're starting to discover, this gets to Giovanni's question, I think, is that, and I mentioned it briefly in the middle, is that there is a directionality because the RNA exits only one portal. And it was really hard to find where did that exit, but there's this one little point. So my question to you is, I wonder, and this is maybe a potential usefulness of this, can you find the tickle point? Like if you hit it here, boom, you can make the thing eject.
I'd love to write a paper with tickle points.
And then the other point that maybe addresses one of Keith's questions is there are at least two other medical importance for this, even this sort of data. And that you mentioned the nanofabrication field. So there has been this really kind of neat idea of maybe using the empty particles for delivering, because they're really, really good at it. So maybe take care of doing that to maybe deliver gene therapy. So put what you want, engineer what you want. And on the other hand, they're trying to develop drugs against those last little proteins that seem to plug up that last pore after they load up the RNA or the DNA. And if you drug those, then they can't let it out.
Yes.
So that's a really, but you have to capture it before you know you've got the virus. So it's sort of a preventative. Anyway, so I think that gives you a little bit of biological relevance to it.
Okay, one more. Well, I'm afraid I'm going from the sublime to the ridiculous.
Let's try it.
Almost pure mathematics. And it's just qualitatively, the stiffness comes from the places where there's curvature, where there's intrinsic curvature. If you have a very thin shell, it's the places where you see pentagons. Mm-hmm. And of course, there are always a dozen of those on if you have just pentagons and hexagons. So that in the Frank Casper structures that have bigger H and K, you get more and more regions that are essentially flat. They're hexagons, and that's the stiff part. That's the soft part. That's why I was getting confused about your coordination of it. But I would think there's some trend that's just qualitatively clear from that observation that the large hexagon regions are not much stiffness at all. And all that. So I'm going to leave it at that. Said it'd be ridiculous.
Thank you. Thank you for the suggestion. But of course, as I said, if you want to have many hexamers, then you need these big things, and then one trajectory is in 4 or 5 weeks. So, but still, one can probe it at shorter trajectories, and maybe one could see what you say. Yeah.
I guess it would practically.
But also remember, but you also remember though that you have in mind this geometrical structure, which seems flat and sharp, these edges, whereas this is actually made of molecules. So locally, it's really hard to see as this end of hexamer and this end beginning of pentamer. I mean, it's a cloud of things,
It should be made clear, the hexagon and pentagons it's just the symmetry.
Right, the symmetry, right.
When you are
Whereas in
shooting
fact you
into
have
real
hexamers and
That's
pentamers.
understood.
Another
Oh, no,
step.
it's qualitatively at some scale that could be. where-
So, but geometric, if it was really a geometrical object, then yes, you are definitely right. But here, since not that, so we're really talking about symmetries, that it may or may not be true.
Okay, we'll continue at lunch, and thank you very much.
Thank you very much.